So good evening. It's so glad to see all of you. I'm Julie Freischlag. I'm the Dean and Vice Chancellor for Human Health Sciences here at UC Davis. And I have spent a lot of time with Jan because this is one of our most amazing programs. And every time I come and listen and hear about what people are doing with stem cells, it's just amazing how we're already affecting people's lives. And I'm really delighted that all of you felt that it was time to celebrate Stem Cell Awareness Day. And tonight's symposium is the first one in the state to showcase this Awareness Day. And of course, we always feel like we're a little bit more advanced than anybody else anyway, so we're glad it's starting here. And I'm really excited to share some of the latest news from UC Davis into understanding and really guide the power of stem cells to treat disease and injury. In fact, we were actually interviewing a new prospective faculty member that would love to come here to be part of this, as his work is in diabetes and revascularization. And where he works in Canada, he doesn't have the resources and the colleagues that we have here. And one of the reasons I came out here from Johns Hopkins about 7 months ago also is because of the UC Davis Institute for Regenerative Cures and the opportunities that I've seen. And Jan was actually in my office, I think, within her first week that I was here. She was getting a grant together and wanted to have my letter and let me know what she was doing. California is leading the way in regenerative medicine. There's no question about that, and research is underway in our state and at UC Davis, and it's highly regarded throughout the nation and the world. Our stem cell director, Jan, who's going to talk to us, always has this amazing vision, and the people that work in her lab, she knows them all. The patients she cares for, they all know her, and she really has this energy for the program, as she is a local hero too, as you know, she grew up here. She's built a top-notch team of stem cell experts that she brings through her lab from all disciplines, and it's so collaborative environment that it really stretches not only for our campus in Sacramento or campus in Davis, and she also taps many students from Sacramento State and has everyone work in the lab with the same energy she has. The novel approaches that have been undertaken include the first cellular therapy for Huntington's disease, and I just met Vicki for the first time tonight, but have met many of her patients and advocates and people that go out to tell the news of what she's doing, and so it's so great she's going to share with us tonight. I know John Laird really well. You know, he's in my field of vascular, and he's doing clinical trials to treat critical limb ischemia and to prevent amputations, which still is a problem, even though we have many great interventions such as balloon angioplasty and bypass. I'm a vascular surgeon. People still lose their limbs. And Dr. Peter Belaski will discuss the rapid progress he's been making to develop a stem cell treatment with people that can't swallow, which has got to be a horrible thing when you can't eat. You'll learn about this work and more tonight because UC Davis clinicians and scientists work side by side. I've been over to the lab to see it, are harnessing the collective wisdom of all of us and expertise to make these breakthroughs for unusual disorders, hard to treat disorders, and they're having successes. What helps set UC Davis apart is the teamwork and the full range of investigators we have here at the campus, as well as UC Davis in Davis. Our good manufacturing practice facility, for example, is one of the largest and most advanced facilities for stem cell testing and manufacture anywhere. And I've been through that too, the part that he let me see. I had to get that of some parts. We also bring together the clinical knowledge, our health system, with expertise for the School of Veterinary Medicine, and that's another huge advantage we have. And I've really enjoyed getting to know Dean Larimore, and I have visited the animal hospital out at Davis. It actually competes quite well with the human hospital here in Sacramento as far as the care and the things. I saw a dog on dialysis as I walked in, and all the care they give. And they have lots of trials. And one of the first ones I saw before when I was deciding to come out here was regenerative medicine to treat dogs, cats, horses, and recover from disease and injury. And these therapies are critical for guiding the development of similar stem cell treatments for human illness and disease. The UC Davis Department of Biomedical Engineering also provides further expertise in technologies and approaching regenerative medicine as well. Everything from finding new ways to monitor the movement of stem cells after infusion to repairing the damaged cartilage in the knee. And we have more important partnerships with industry. We know that private industry and fellow academic institutions, we have to partner with everyone to get the resources together and expertise to make advances. And we do that not only in the United States, but also throughout the world. This work would not be taking place, however, if it wasn't for the voters of California and the state stem cell agency they created 10 years ago. And we heard about that when I was back east. We were very jealous, and we really were impressed with the passage of Proposition 71. In fact, if you walk by our Institute for Regenerative Cures, which is just down the street, that you'll see there's a sign there. This is a facility supported by the California Institute for Regenerative Medicine. We'll never forget that ability to get started. CIRM has been a catalyst for so much of the work now underway at UC Davis, and its early support enabled UC Davis to open the state's first CIRM-funded research laboratory and helped us launch dozens of stem cell studies and clinical trials. And we're so honored to have the chair of the governing board of California Stem Cell Agency, Jonathan Thomas, here tonight, and he'll talk right after me. Chairman Thomas, or JT, as I was introduced to him tonight, is affectionately known around his office, has brought this unique blend of expertise to CIRM. He's a lawyer, an investment banker, and a biologist. Didn't think you could put all those three together, but they also never thought a surgeon could be a dean either, so we all outside our norm. And he's a person equally comfortable in government as well as financial and scientific worlds. And that's so important these days that we get everyone together to advance the care of patients. He helps guide the funding decisions of the agency, a 29-member board, which is charged with distributing $3 billion to fund the development of stem cell therapies for people in need. Chairman Thomas also has a long-standing commitment to patient advocacy, having spent many years on the board of an organization now called Ability First, which assists children with spinal cord injuries and mental disabilities, youngsters who could benefit from stem cell therapies. Like UC Davis, John Thomas is working to turn stem cells into cures. So I want to welcome Chairman Thomas, and thank you very much for being here tonight.
Thank you very much, Julie, if I may call you Julie, since we're into informality here, and welcome everybody. It's a real privilege for me to be able to come speak to you about CIRM, this grand experiment that the voters passed 10 years ago. This is our 10th anniversary coming up in about a month. We plan on having an event to that effect near our headquarters in San Francisco. CIRM, for those of you who aren't familiar with it, is a creation in California, and only something that California would ever come up with. I'm sure Julie watched in other states as similar things were tried, where it was put on the ballot to create a basically autonomous agency to fund medical research with public dollars. Nothing like it in the world. And what it did was it transformed California in authorizing the issuance of $3 billion of state bonds into it. California became the 800-pound gorilla in the world in the field of stem cell research. And indeed, we've had many senior stem cell scientists from all over the nation and the world move to California just to have the opportunity to apply for funding for their work. I think by all accounts, stem cell technology, regenerative medicine in general, which includes stem cell work, it includes, you folks may have heard the term genomics, which is the sequencing of all the DNA in your body using very fast computational techniques that are getting further and further refined to identify mutations in your body that you can then use as the basis for personalized medicine techniques that include stem cell work. It includes gene therapy. Sometimes it's a combination of the two. All of these are the sorts of things that we're funding. Of interest, I think, when we began, the field of stem cell research was in its infancy. The first human embryonic stem cell was isolated in 1998, and everything in the cellular therapy space has flown from that. And as CIRM started in 2004, really 2007, because like any good novel thing, we were sued immediately upon passage. It took about 3 years to get through all that, which we did, and we basically had the kitchen sink thrown at us, and it was actually a good thing because it established that, in fact, what we were doing is legitimate. Believe it or not, Prop 71 put the right to do embryonic stem cell research into the California Constitution. So it's something that cannot be changed unless there's another ballot measure that takes it out, and that's not going to happen. California is very progressive, always has been. California is very cutting edge, and it's that sort of frontier spirit that came out in the passage of this. So, okay, so what have we funded? Well, there are, as you know, any number, unfortunately, of currently incurable diseases and conditions. CIRM to date has funded research in 39 of them, including some terrific projects that you're going to hear about here at UC Davis. Our funding has gone towards research in all the biggest diseases: cancer, diabetes, heart disease, neurological disorders, vision impairment, all the way down to a bunch of rare or so-called orphan diseases. In each instance, we have funded typically teams of scientists at various either medical schools or, such as here, or research institutions. You've heard of the Scripps Institute and Salk Institute and the Gladstone Institute. And we as well fund work at biotech companies. Key is, it's all in California. So $3 billion going into research only in California in 39 currently incurable diseases and conditions. And I think last count we're at about 67 different funded entities of the three kinds I just mentioned. Nothing like this anywhere in the world. Okay, so where are we in our research? So in the earliest days, because the field's new, we funded basic research. We funded a lot of infrastructure, construction of stem cell institutes around the state, and we funded training. And as things progressed over the years, the research sort of worked its way through the pipeline, and after basic research, we have what we call early translation. So you find something in basic research, what do you do with it? Well, you move it on, refine it, and try to get it into preclinical work, which is a fancy term for research in animal models. If you get through that and you convince the FDA that your results are compelling enough, you then go into human clinical trials. And that's sort of where the biggest awards have focused in the later preclinical days and the clinical trials themselves. You'll be, I think, impressed to know that our biggest awards, which are by medical research terms huge, we call them disease team awards. You have a number of them here at UC Davis. These are up to $20 million awards. Now, for those who aren't familiar with medical research, you know that the basic driver has always been NIH, and the bread and butter award of NIH, their R01 grants, average $280-something thousand. These are $20 million. These are gigantic. And what they do is it allows for institutions to bring not just folks in-house, but to combine institutions to bring, and in some cases, some of these teams are institutions plus companies. It brings all the expertise to the table that you need to tackle the many complex problems in this fledgling, developing field of cellular therapy. I'm very proud to say that our first round of disease teams were awarded in 2010. We had 14 of them. We've had 2 subsequent batches. I think we have 30 some odd disease teams altogether. Of that first group, 9 of those by the end of this year are going to be in clinical trials. We actually have one from the second group, which is sort of an unusual one, which had had the first phase of its clinical trial funded by NIH. We're funding phase 2. So at the end of 2014, we look to have 10, possibly 11 in total in clinical trials. For those of you familiar with pharma and their batting average of how many things they start funding research and development on and how many make them to clinical trials, much worse batting average than that. This is pretty cool, and it's a testimony to first our peer reviewers who are drawn from all over the world to evaluate grants, but most of all to the talent of the scientists here in the state. This is top drawer and world class in every respect, both with the scientific research. We have here at UC Davis, a very critical element is the manufacture of the drug, the product. Gerhard over here, who is a master at that, one of the world-renowned people in that field. You bring different elements to play. All of this has to go into making the product. So just to give you, I'll give you just a quick example of one of these things that's in clinical trials. We have a company in San Diego in the field of type 1 diabetes called ViaCyte. Type 1 diabetes, for those of you who aren't familiar with the distinction, is an autoimmune disease where the person's immune system actually destroys the beta cells that come from the pancreas, that are the ones that produce insulin to break down the blood sugar in the body. So ViaCyte is taking embryonic stem cells, and cool thing about these things is you can program them to become other things, and embryonic stem cells become everything in your body. So in this case, they're programmed to become sort of the halfway version of beta cells that produce insulin in the body, and those, they call them progenitor beta cells. And those progenitors are put into a little capsule. It looks like a flat little envelope that is about that big, and it will go underneath the skin, and it is porous, and it's big enough to let the blood sugar molecules in, and big enough, the pores, to let the insulin out, but not so big that the big nasty T cells in your immune system, which are destroying the beta cells, are able to access the cells in the envelope. So basically what you're doing is you're creating an artificial mini pancreas that's going to crank out insulin, and it's been used in animal models where they humanize mice to have the analog of human diabetes, and in the mice it totally cures the diabetes. That just got approved for clinical trials within the last month or so, and they are now engaged in actually enrolling patients. That's one example. I could spend about 2 hours giving you a litany of projects, some of which you're going to hear about tonight, that are so cool and so cutting edge. And as we sit here, it is science. You don't know what's going to work and what isn't, but everybody in the field is really jazzed about this stuff and feels that some of this stuff's going to work. And this is, I refer to it as big game hunting. This is Jonas Salk going after the polio vaccine, all these things. So anything that succeeds will change the world with respect to that disease or condition, which is what makes the field of regenerative medicine so neat, so compelling, such a great thing for the medical students to be involved in. This is where the action is going to be, certainly one of the big areas. And I believe that 20 years from now, we will look back at this era and genuinely see, when a lot of this stuff is by then routine, that we were all part of a real revolution, all courtesy of this grand experiment that is CIRM. So thank you for the chance to come talk to you guys. I appreciate it. Keep up the great work you're doing here, and we look forward to lots of good stuff coming out of UC Davis and coming out of California for the benefit of its citizens and indeed for the world. So thank you.
So thank you, Chairman Thomas. What a wonderful thought for type 1 diabetes. And we appreciate the time you've taken to be with us for Stem Cell Awareness Day. And look forward, we'll get together, have lunch, and figure out where we're going to go next to do that. So now I'm really pleased to introduce our next speaker. Fred's the Vice Dean, UC Davis, my right arm, partners in crime, and he really, one of his most amazing qualities is putting together teams. We had a meeting today at UC Davis with our provost and our vice chancellor of research, where we, along with him and Dr. Berglund, discussed what we're doing presently in research, how we evaluate research productivity in our faculty, what kind of people we support, and what are the things, where are we going? And one of the big things we talked about was team science, which you are. Looking at great big teams to solve problems, no longer one person in one lab thinking by themselves, and looking at where those teams should be. And frankly, we talked all about your space, Jan. Your ears were probably burning about how to have them give us lots of money to help us do that. We didn't quite get that, but how those big teams that she's assembled will be great. Fred does that in our office, putting teams together, genomics, stem cells, and he is on one of the grants for the CIRM Center dedicated to training stem cell scientists. Because the other thing that turns Fred on is getting young people excited to go be the new academic physicians of the future, to be the vice dean or be the dean or run the stem cell center, because someday we do need to retire or we need to leave, one of the two, to do it. And he's really incredible, encouraging students and fellows and junior faculty to really look into the field of regenerative medicine to do exciting things like JT just said. He knows the importance of academic skill development and looking at that, and especially in this field of stem cell science. And he's a mentor and a teacher. We were just talking in my office today because we both have this thing. We just come and teach because someone taught us. It's that simple. And so that's what we do. We just teach. And I'm really glad to be working with him so closely. He makes everything make sense to me and has great big ideas. So I welcome Dr. Meyers to the podium.
Thanks, Dean Freischlag. We're a great team. We love working together. So let me just leave you with one message tonight, and I'll sit down so you can get to the really exciting part. I know many of you, we've seen each other over the years. We've seen our program grow and change, haven't we? And we were able to convince Jan and Gerhard to leave Missouri. It was tough. They love Missouri. They had roots there. You still hear the Missouri accent when Gerhard talks. I think that's a Missouri. Pretty sure that's a Missouri accent. But they came on a dream, because there wasn't much here, was there? But we invested university money, and we invested CIRM dollars, and we put together the institute. We didn't ask you to invest because it was a dream. It was betting that something good would happen. But we took a lot of university money and a lot of CIRM money and a lot of sweat equity, and I think we've built a pretty fantastic team that is leading California and the nation in what Dr. Thomas was talking about in terms of translating stem cell biology to new therapies. So now I am back here to talk to you. We can't do what we want to do in team science, in creativity, in mixing people up, and in really breaking down barriers. We can't go to the next level without your support. I was really happy this morning to sign a thank you letter to a couple from our region who donated a six-figure gift to Dr. Nolta. All the other stem cell centers in California have names on them. The Broad Institute for Stem Cell Research. Well, we need that here at UC Davis now. Now is the time. Now you're not betting on a dream. Now we're betting on something we got. It's not on it might happen. It's happened. But now we need to go to that next level. We need to really continue to recruit, train, and as Dean Freischlag has said, develop the teams of the future. And so we now need Sacramento to step forward, not just to have philanthropy for a soccer team, which I think is really cool, by the way, or the Kings. We need philanthropy for the UC Davis Institute for Regenerative Cures. We need legacy gifts in your estate planning. We need gifts that will support our junior people, our students. Dr. Nolta and her team have students that go all the way from high school to me. And I'm the most difficult student. But it can happen. And we do mix and match. We can recombine people from the vet school and the med school, from biological sciences, and out into the community. These are not what most people do, but Dr. Nolta and her team have a vision that bringing people of diverse backgrounds and diverse thinking together will result in something very special. You're going to hear from my colleague Peter Belafsky, who recombines with the vet school and a remarkable number of people who do things that are unbelievable. I heard him give a talk last month about it. I'm still amazed at what he's doing. But that's not easy to do, and that's not what we get NIH dollars or CIRM dollars for. We get those dollars for the sure thing, or the almost sure thing. Philanthropy drives innovation. It allows us to bring people together and ideas together that are indeed innovative, the big, high risk, high gain ideas. We can cure AIDS. We can cure Huntington's disease. We can do that. We can prevent amputations. We can do that. But that won't happen with grant money. The university will still invest our money in this program. Dr. Freischlag and I are committed to investing our dollars, a significant portion of our dollars, into this type of activity. But we need you to partner with us. We can't do it by ourselves. Estate gifts, real dollars, and asking your friends and your acquaintances to come and learn about us and think about how your investment can change the future of California and, most importantly, for the patients and families that we take care of. So it's 734-2412. Give me a call. It's FJ Meyers at ucdavis.edu. We'll go anywhere and talk to you about this. You can have a tour. We can have a one-on-one conversation about what your passion is. We can go and meet you in your home. We can have lunch. But I am committed to this. Dr. Freischlag is committed to this. Our Associate Vice Chancellor for Development, Chong Porter, sitting right there. I'm going to get a grade from her tomorrow. It's going to be another C-. I'm trying to do better. We can meet you where you want to go, but come with us, because I know many of you have been with us since the beginning. You've been here for years, and now it's time for us to really go to a completely different place. So call me. It is 734-2412 or FJ Meyers at ucdavis.edu, and we will come and chat with you. And we will talk to you about how we can continue to be partners, because you are our partners. You've been with us for a long time. Let's continue to do this together. That's my message for tonight. Happy Stem Cell Awareness Day. Thanks, Dr. Nolta.
Well, thank you so very much, Julie, Fred. Thank you for all of your support. It's incredible to be here, and so many of you have been here from our very first stem cell dialogues until now, and glad to see you again. I am Jan Nolta. I'm the director of the Institute for Regenerative Cures, which is a big building and a big group of people, and the stem cell program, which is an even bigger group of people that spans both campuses. And I'm just going to tell you a little bit about it tonight. And Dean Freischlag, I know you have to go because you have a guest from out of town. So, and thank you both for being here. So we have this wonderful building, and this is, as was mentioned, a facility funded by the California Institute for Regenerative Medicine, or CIRM, that JT talked to us about. It's a huge building, 109,000 square feet. It was a historic building, part of the State Fair, and funded by a $20 million gift from CIRM, grant from CIRM. We now have over 200 scientists, students, veterinarians, and MDs working together, and you'll hear about some of those projects tonight. I'm just here to introduce my wonderful colleagues. And this is really what's so important to us, developing new therapies where conventional medicine stops and doesn't have further options. None of our doctors who work here want to tell their patient, I don't have anything else to help you, whether it's a veterinarian or a human doctor. We need new therapies. And so that's what we're all working on, and that's what we're hoping for regenerative medicine and stem cell therapies. And they are really beginning to come to fruition. So this is a bunch of numbers. I'm just going to go through it really quickly to give you an idea of where we've come in the last few years that we've been working on this. We currently have 150 affiliated faculty members. We have 16 of these disease teams. You'll see a nice graphic about those in a minute. Many academic and industry partners. We've had $125 million in the state-funded grants from CIRM, funding for facilities, team training, research, and for these early phase clinical trials that you'll hear about tonight. And this is huge. This is, as JT mentioned, these big grants that really allow us to go forward into the clinic. And with the philanthropy that Fred was just begging for, sorry about that. We do have to give him a grade tomorrow, Sean. With the philanthropy, when we get small amounts of seed funding, we're able to start the really novel, cutting-edge things that we don't have any data for that allow us to go forward for these big grants. So we're extremely grateful to anyone who has donated to us. We administer a very cool thing. It's the upside down, sorry. It's the statewide umbilical cord blood collection program. And this is really neat because cord blood after babies are born is usually thrown away. But through this program, it can be collected and sent to public cord blood banks where it can be used to save lives. And so we run the state program that is doing that. Very, very proud of that. We've generated many jobs and construction-related jobs. And this was very important because the money from CIRM came to start building right when the construction industry had really tanked in about 2008. And the people working on our building were extremely grateful for the work. So this was very important to us. We have 10 adult stem cell and regenerative medicine clinical trials currently ongoing or just completed. And to me, that's really incredible. I mean, I've dreamed of this my whole life, and now we're doing it. And we have 18 in the pipeline, and you'll hear about 3 of those tonight. And we have this wonderful good manufacturing practices facility, which is where we manufacture the cells for the clinical trials. My amazing colleague, Professor Gerhard Bauer, who's here with that Missouri accent, although he's really from Australia. Austria! Sorry, how could I get that wrong? Yeah, so many places, yeah. And we do what are called IND-enabling studies for the FDA, for the Food and Drug Administration to start these novel trials. We have cores with recharge rates where companies can come in and do their research through us. And we have a lot of fee-for-service contracts with a lot of academic and industry partners worldwide. And these are really our bread and butter contracts that let us keep our people with paychecks. So very, very important. And I just wanted to show this on behalf of Professor Bauer. This is his wonderful UC Davis Good Manufacturing Practice facility. It's really a clean room facility. We grow our little cells that are going to come over to the clinic and be administered to the patients. This is a multi-layer cell hotel, and the cells are growing on different layers in here, and so it's like an apartment building for the cells, and they grow on each layer, and they grow in the fluid like a little aquarium. And it's very important to us to grow these in a way that's very sterile and reproducible so that every clinical trial that we do, we can expect the same outcome for the next patient. Types of stem cells. So far, we're working with adult stem cells, but we do have some pluripotent stem cell therapies waiting in the wings, trying to move those forward. The adult stem cells come largely out of our bone marrow and even from our fat and from other tissues. And we have the hematopoietic or blood-forming stem cells. So these are the original stem cells that have been around for 50 years through bone marrow transplantation and now cord blood transplantation. That was the first stem cell therapy. And now we work with these mesenchymal or supporting cells. We call these the paramedic cells. They really help other tissues heal. And you'll hear a lot about those tonight. These embryonic stem cells have been around for about 12 years now, and they, as JT had mentioned, make all of the cells in the body, like those new pancreas in a bag that the company Viaside is going to be starting. And that's a really exciting trial. But we also have an even cooler pluripotent stem cell that can make any cell in the body. And these are induced pluripotent stem cells, and those come from the patient's skin. We take a little tiny sample of the patient's skin. It's kind of like getting your ear pierced, but on your arm. And we can grow out the cells. We kind of turn them back in time so that they're just like the embryonic stem cells. We can then differentiate these into neurons or muscle or liver or kidney or whatever we want to, whatever organ we need to make from them. And that is really exciting technology, a lot of that going on. in our institute. As I'd mentioned, we have a lot of these therapies in the pipeline, and so we go from basic research through the translational research, preclinical research. A lot of the teams that you'll hear about tonight are mostly in this area, just getting ready to start a phase 1 clinical trial. And then these are the clinical trials that we have ongoing. And then there are approved therapies for stem cells and regenerative medicine that we use in the hospital all the time. So bone marrow transplant, organ transplant, and also in the near future, making organs better through perfusing the patient's own stem cells into them to kind of revitalize them when they come from another donor. That's something that is a wave of the future that's coming up. Skin expansion and grafting, the cord blood transplant, and cells and matrix for wound healing. And these are all very exciting stem and regenerative therapies that are ongoing and are approved. And by approved, I mean the insurance reimburses it. And so that's where we hope to get with all of this basic research and the translational. We hope to get the product through so that it becomes a accepted standard of care, and it will be reimbursed by insurance. And then it can go to every patient who needs it. So this is my wonderful daisy of our different teams that Jane made me. Thank you, Jane, and it's wonderful. And these are all of the teams with which our stem cell program is working, most of the teams anyway. And you'll hear about some of these tonight. And you can see that we're really working in a lot of different areas and trying to make a difference for patients who don't really have other options. And we work with a lot of different centers here and departments and other programs. And this is teamwork at its finest. This is how everything gets done. Nobody knows everything, but as a team, we can know a lot of stuff and we share our expertise. And so it's really great to have all of these opportunities. Partnership really is the key here. We rely on leadership, industry, community, and education, and we do a lot of training of students. And these are a couple of our high school classes, and the future of medicine is very important to us, training the next generation. And so with that, I'm done with my little introduction. Please come take a tour if you haven't already, and see us down at the Institute for Regenerative Cures. It's down at the end of Stockton by Jack in the Box. That's our claim to fame. And we love to have visitors and to give tours. And now I'd like to invite up my awesome colleague, Dr. Vicki Wheelock. And she's the director of the Huntington's Disease Clinic and works tirelessly and has so much passion to make a difference for her over 300 patients that have Huntington's disease and juvenile Huntington's disease. And it's just incredible to work with her. So, Vicki, come on up.
Well, thanks so much, and thanks to everyone for coming out tonight for Stem Cell Awareness Day. It's a real pleasure to see you, and it's also a pleasure to see JT from CIRM, and to see our deans, and to follow Dr. Nolta, who's our rock star of stem cell science. And I want to thank you in turn for being here, thank the university for recruiting you, and Dr. Bauer to come here so that we could start to do the type of research that could make a difference for our patients and families. So I'm going to give you an update about a grant, a disease team grant that was funded back in 2012 to develop a treatment for Huntington's disease. And the disease team grant actually funds the preclinical research in the lab working with the animal model of Huntington's disease, which is a mouse model with the human Huntington's disease gene, all the way forward to getting FDA approval with something called an investigational new drug license, and then into patients where we plan to do a phase 1 early phase safety trial to see if our treatment is safe and can go forward as a treatment for Huntington's. So Huntington's disease is a devastating disease. It's a neuropsychiatric disease. It's slowly progressive, it's fatal, and it passes from generation to generation. It's truly heartbreaking. And even though a relatively small number of people in the country have Huntington's disease, only about 30,000 people, there are at least 150,000 people who are at risk of getting this disease. And we think that the costs for Huntington's disease approach over $2.5 billion per year because people get sick at the often at the peak of their lives when they're earning, they're having families. sadly, we see juvenile Huntington's disease, and we even see it sometimes very late in life. And the key symptoms are loss of motor control with involuntary movements that can be very severe, cognitive loss, and very severe psychiatric problems, all bundled together. So the disease passes from generation to generation, and here's the low-hanging fruit about Huntington's disease. We know what causes it. There's a specific gene. Everybody who has Huntington's has a mutation in this gene. We all have Huntington's genes, but people with Huntington's disease have a mutation in the gene that causes an expansion. There are too many nucleotides in a row in a stretch of the gene that leads to a protein that is malformed and malfunctioning and leads to cell death within the brain. So Huntington's passes from generation to generation. Normally there are about 20 or so of these CAG messages together. In Huntington's there are greater than 39 or 40, and sometimes we see 60 or 70. That's what we see in people with juvenile Huntington's. The mutation is present in every cell of the body, but it only seems to affect the brain cells and only certain brain cells. But the brain cells it affects are deep in the brain in an area that controls movement and thinking and behavior. So a specific neuron called the medium spiny neuron gets sick and dies out. And over time, we see progressive death of the cells in the brain until people become disabled and bedbound and die. So the strategies now for Huntington's disease, we only have palliative care. We can only help people manage the symptoms the best that we can, but unfortunately they get worse and worse over time. So we and others are looking at neuroprotective therapies. Can we find a way to help delay the onset of Huntington's or slow the progression, keep people in the early stages longer so they can stay healthy longer? Sadly, there are two studies that have been going on for many years now, NIH funded, that we hoped were going to help to slow the progression of Huntington's, and they have both failed. And so hundreds of patients were enrolled around the country in these studies, hoping that the drugs they were taking versus placebo were going to help them, and sadly, they're failures. So the funding has been stopped, the studies are ending. We need to have more studies. Even more tantalizing would be a cure for Huntington's. Why not go all the way and find a way to cure it? And we think that because we know about the gene, we know about the mutation, we know about the mutant protein, that there must be a way to switch off this gene. There must be a way to switch off the production of this protein that's killing the cells. And there are a number of different lines of research that we think might be able to do that. And we think that our own research will lead to breakthroughs in this area in the future. So Dr. Nolta's team developed the concept of using these mesenchymal stem cells, the body's paramedics, to engineer them because they can actually be designed with gene therapy to produce factors that are needed to help treat diseases. And so this is a review that she published a couple of years ago looking at using these cells as a proposed treatment for Huntington's disease based on the science in the cell culture in the dish, but also in animal models that have been going on for many years. And she identified a compound called BDNF, which is a growth factor, a trophic factor like fertilizer, I think, to help out brain cells. And it turns out that the Huntington's mutation causes the levels of this factor to plummet so that the levels are very low in the brain. And we know that when those levels are brought back up in cell culture and in animal models, that helps to rescue the sick and dying neurons. It helps to restore the neurons back to health. One of our collaborators in Michigan, in fact, did a study with a Huntington's mouse model showing that if you gave these cells mouse stem cells that were engineered to express this protein, that it would help to rescue those nerve cells and help the mice to have a slower progression of Huntington's. Not a cure, but to make a significant difference in the progression. So the model here is that the cortical neurons on the wrinkly part of the brain, the outside of the brain, are actually delivering BDNF down to the medium spiny neurons all the time, every day. In the presence of the Huntington's mutation, BDNF is blocked and it can no longer reach those cells and help them to be restored, and the cells get sick and die out. Our goal is to engineer these cells, the mesenchymal stem cells, and implant them in the brain to deliver BDNF, and we hope that this will help to rescue the sick and dying neurons. And we know from various lines of research that BDNF can also recruit new cells to this area of damage. So we hope that both of these mechanisms will help to slow the progression of Huntington's. So this is the name of our grant. MSC's engineered to produce BDNF as a treatment for Huntington's disease. And now you know what all those words mean. And the goal of this study is to go forward into a phase 1 trial, an early phase safety study in people with Huntington's. And you know now that we're using mesenchymal stem cells that are engineered to do this. The plan for this grant is actually a 4-year plan. It's very fast to do something like this. We have a preclinical phase and we have a future planned stem cell trial. The preclinical years, we're in year 2 right now of this grant. Many studies are going on in the Nolta lab looking at the mouse models and working on getting this ready for going forward to the FDA. And I'll say a few words about those. In the meantime, we're doing a study called Pre-Cell. And Pre-Cell is a study for people with early stage Huntington's who might be candidates to go forward to the stem cell treatment that we plan to deliver. And we're now into the 2nd year of that study. We have a number of people who come and see us every 6 months, and we do intensive evaluations for those patients to measure how their Huntington's is progressing, to do brain scans. We do spinal taps. We do all sorts of things to characterize what's going on with their Huntington's so we can be ready when we have that investigational new drug license to go forward into patients. This is a diagram showing the organization. We have 2 big teams that are working together. We have to coordinate the science and the medical care for the patients. And we have a number of consultants. Many of them are here at UC Davis. Many of them are consultants that are far away. We have consultants in Australia, in Boston, in Seattle. We have people who've been advising us to make sure that we can do the best possible science. So the preclinical studies, I told you I'd share with you a list of things that are going on in the lab right now. And these are things, some of which have been completed and are ready to go. Others are still underway. The MSCs have been engineered to produce BDNF. Boy, do they produce BDNF. We can measure those BDNF levels. We can ensure that those cells are stable after they've been gene modified. They don't do something unexpected or unsafe. They're being tested in mouse models for safety to make sure that they're safe. And right now, we're also doing studies to see how long they survive in the brain once they've been implanted in the animal models. We're measuring the effectiveness of these cells in the animal models of Huntington's disease, and all of this is going forward to put together a package, all the pieces of the puzzle that we need to go forward to the FDA to get their stamp of approval to go forward into patients. So this is a vector map. This is actually a map showing this amazing work that's been done in the Nolta lab to engineer these cells. And if you look around 8 o'clock, a little tiny writing right there, right around 8 o'clock it says BDNF, and that's the stretch that's going to be making BDNF. And right around 7 o'clock is the promoter to kind of get that going on. So very complicated, very elegant work. This is actually showing the BDNF production from the cells. And so the team is measuring how much BDNF is being eluted by the cells per number of cells. And it depends on how much the vector is introduced into the cells. We can increase that, and they're pumping out BDNF beautifully. These are some pilot studies in a small number of mice. Pilot studies are sort of initial studies in small numbers of animals or over a small period of time that show that when we give this treatment to the transgenic Huntington's mouse, the mice get better, their coordination is better on a coordination test, which is sort of like a log rolling test that they do in the lab, versus our control mice that get just a saline injection. And we can also look at the brains of the mice after they die and see that these cells survive up to a point that they actually are inducing growth cones for new neurons, just even in a very short period of time. We can show the biological effect of these human cells implanted into mice. So currently we're carrying on a large number of studies. These are called efficacy studies, and efficacy means to see if it works or not. So those studies are actually being done in 2 different mouse models of Huntington's disease, and we're doing behavior testing. We will be looking at their brains after they die. We're doing very detailed analysis of the BDNF. We're doing very detailed analysis of what the BDNF is doing to help restore those cells and bring new cells. A large number of experiments are going on right now. Some are finishing up, some are just starting. We plan to have these done by the first quarter of 2015 to have our package together to get ready to go forward to the FDA. So I just in the last few slides want to update you about what's going on in the clinic. I mentioned we have a study called Pre-Cell, which is going on right now, where people with early stage Huntington's have volunteered to come into an observational study where we just check them. There's no treatment. And in the future, we plan to do a study called HD-Cell, which will be the stem cell trial, the treatment trial. So the primary objective of Pre-Cell is to enroll somewhere between 26 and 40 patients with early stage Huntington's to do these types of assessments every 6 months over time in support of this future trial. We anticipate asking the people in Pre-Cell if they want to go forward into HD-Cell when we get there. And so we'll have a very good baseline for them to help us make sure that it's safe and help us measure whether we might be helping out with their Huntington symptoms also. We have a large team of experts assembled, again, mostly from UC Davis, our team right here, but also from our consultants and other far-flung labs who are world experts in Huntington's disease research. And we're really proud. I wish I had time to tell you all the things that we're doing. This is a list of inclusion criteria. There are 6 inclusion criteria: people with early stage Huntington's who are otherwise healthy, who have stable psychiatric symptoms, have a companion who can help us keep another set of eyes on them. And here's a list of some of our exclusion criteria. It's as long as my arm. And there's a place you can go to on the web to look at this. If you put in Pre-Cell, you can get into the details about the inclusion-exclusion criteria. This is a table showing how the patients are coming in every 6 months, all the different tests that we're doing. These visits last over 2 days. We can't even do all of them in one day. And then we call them every 3 months in between to make sure that their health is stable. So far, the study was approved a year ago. We have screened 27 patients. As of 2 days ago, we've enrolled 22 people in the study so far, and we're doing these detailed examinations. It's going forward very well. I just wanted to show you a bit of the data from the study. This is actually an image, a composite image of brain scans that we've collected from people in the study. And this was analyzed in the DeCarli lab. Dr. DeCarli is our Alzheimer's disease expert here at UC Davis. And so his team was able to assemble the scans and to actually compare those scans of the patients with Huntington's to a normal scan and a normal aging individual. The areas in the blue, the cooler colors, the green colors are areas where cells are dying out in Huntington's. Even in early stage Huntington's patients who are still working, still raising their families, we can see a lot of change in the brain already. The areas that are red are where the Huntington's patients have extra cells, more cells, because they're younger than the people who are in the composite for the control. And we can see this very, very clearly. Those are the areas that we're going to target with these stem cells. So the plans going forward are to continue the observations in Pre-Cell, to continue and complete the studies in the mouse models, and to go forward for that IND with the FDA. This is what our therapy will look like. We'll be implanting these cells through brain surgery into the brains of the patients who have Huntington's disease. They'll be awake during the surgery. They have a little tiny, what's called a burr hole drilled in the brain, and the brain doesn't have any sensation. It doesn't hurt to do this. This type of surgery we do all the time for Parkinson's disease to implant pacemakers and things like that. We're using the same sort of technique that we'd be using for Parkinson's disease. All right, and I just showed you where we're putting the cells. Those are the targets of where the cells are sick and dying, and that's where we need to target with the stem cells. So I'll end here just showing a list of the wonderful people in the lab, the wonderful people on the team. It's a huge team. We have consultants who are helping us. It's been very exciting. I am so excited about where we are right now and where we're headed. The grant has enabled us to make huge progress in bringing this science forward, and I hope to be able to tell you next year on Stem Cell Awareness Day the good news about the progress that we're going to make in the following year. So thank you for your attention.
It's an amazing project, and it's wonderful to be a part of it. So next I'd like to bring up my wonderful colleague, Dr. John Laird. He's the medical director of the UC Davis Vascular Center and a professor of medicine. And it's really been my pleasure to work with him on developing a very novel clinical trial that he'll tell you about today. Thank you.
Well, thank you very much, Jan, and good evening, everyone. It's great also for me to be here with you tonight and to support Jan, for whom we all have great affection as both a friend and a collaborator. And it's really been a wonderful partnership for us in the UC Davis Vascular Center, my colleagues, Nassim Hedayati and Christy Pfeiffer and the other vascular surgeons and vascular medicine specialists, to be able to work with Jan's incredible team at the Center for Regenerative Cure is really an amazing group of researchers. And to be able to work with Gerhard, the famous Austrian, Missourian researcher, it's truly a blessing. And we've also established a great collaboration with a group in Spain at the Hospital Reina Sofia in Cordoba, Spain, where they already have a very broad experience with stem cell therapies for patients with peripheral artery disease. And this is our target, peripheral artery disease, or blockages in the arteries to the extremities that can lead to pain in the legs when people walk, a detriment in their quality of life, and when it becomes very severe, loss of limb. And this is a very common problem. Conservative estimates are that anywhere from 8 to 12 million Americans suffer from peripheral artery disease. And as we have a more aging population, as we continue to have this epidemic of diabetes, the prevalence continues to grow. It's been estimated that anywhere from 20 to 25% of people over 70 years of age have peripheral artery disease. And for those with diabetes over the age of 50, as many as a third of those patients will suffer from the complications of peripheral artery disease. And we're particularly focused on patients with most advanced forms of peripheral artery disease, also known as critical limb ischemia or CLI. And these are patients that have such severe reduction in blood flow to their lower leg and foot that they develop gangrene or ulceration on their toes and feet, which cannot be healed without it somehow improving blood flow to that foot. Many diabetics that we see have very severe peripheral artery disease involving the smaller arteries below the knee. So if we look at this slide on the left, we can see what normal arteries look like. And then if we look at the slide on the right side, we can see what a typical diabetic with CLI looks like, where you have basically total occlusion of all of the arteries below the knee. And you just have these squiggly little collateral vessels that are allowing for some trickle of blood flow to the lower leg and foot. Sorry for the shock and awe, but this is basically my life. These are the patients that we see in the hospital and in the clinic every day. Patients who have gangrene, the toes because of reduced blood flow, or the severe ulcers in the bottom of the foot that will not heal without some restoration of more normal blood flow to the foot. And these patients are at great risk for loss of limb without some therapy to improve that blood flow. So what are the traditional options for the treatment of PAD and critical limb ischemia? Well, believe it or not, even in 2014, many patients end up with primary amputation as their treatment for critical limb ischemia. Many of those patients have an amputation even without any evaluation for vascular disease. There are no good medical therapies, and if revascularization cannot be performed, most of these patients will go on to lose their limb. I, as an endovascular specialist, perform angioplasty and stenting procedures on many of these patients, and bypass surgery is still the gold standard in many cases of patients with peripheral artery disease. But in a significant percentage of cases, angioplasty or stenting or bypass surgery is not an option, and these patients will go on to this god-awful complication of below-the-knee amputation if blood flow is not restored to that lower leg. And once an amputation is performed, that just really starts this downward spiral of events that often leads to early death in as many as 10% of cases within the first year and 30% of cases within the first 2 years. And once the one leg is amputated, as many as 15% of patients will require amputation of the other leg within 2 years. And that's often a precursor to death, because once that happens, there's really very little reason for many of these patients to go on living. Despite all of the great therapies that we have, including surgery and angioplasty and stenting, Over 180,000 amputations are performed every year in the United States. At least half of these amputations are due to vascular disease or diabetes. The costs to society are staggering. In 2009, hospital costs associated with amputation were over $8 billion. For the individual patient who has to undergo amputation, the cost for them over their lifetime is about $500,000. And only about 25% of these patients will go on to walk again with a prosthesis after amputation, and less than half of them will be able to go back to work. So the costs to society are really staggering. So the imperative is for us to find an alternative form of revascularization for these patients who are at risk for loss of limb, particularly those patients who are not candidates for the traditional treatments that we perform. So there's been great excitement over the years surrounding this concept of therapeutic angiogenesis, where basically you try and grow new blood vessels to restore blood flow to that deprived limb. The early idea was to give a vascular growth factor directly into the artery or into the muscle of that leg. That evolved into giving the gene therapy, where basically you instill the gene into the artery or into the leg, and that gene would then encode for that vascular growth factor to try and grow these new blood vessels. And then more recently, there's been great interest in cell therapies or stem cell therapies to try and grow blood vessels and enhance blood flow to the lower leg. So what we're trying to achieve is what is seen on this slide. And this is actually a slide that was sent to me by our colleagues in Spain. On the left, we see a patient with very severe disease in the lower leg, but with some collateral vessels, diminutive collateral vessels. This is after stem cell therapy, remarkable increase in the number, size, and robustness of these collateral vessels to the lower leg. So this is the holy grail. This is what we're trying to achieve. Now, the early trials for which I participated in, some of these trials looking at these growth factors or these gene therapies for critical limb ischemia, they showed promise, but when they were ultimately tested in larger clinical trials, they failed. In large part, probably because we couldn't get enough of that growth factor into the area where it was needed, and it didn't stick around long enough to be of use. And so subsequent to these trials, there became a lot of interest, or arose a lot of interest in stem cell therapies for critical limb ischemia. But Jan's idea was really the best idea of all, which was to combine the best of both worlds, basically take stem cells and then bioengineer them to secrete vascular growth factors. And that was sort of the premise behind our disease team project, was to take mesenchymal stem cells, bioengineer them to secrete vascular growth factor, vascular endothelial growth factor, to try and greatly enhance blood flow to the lower leg in patients with critical limb ischemia. And thankfully, we were one of the disease teams that was awarded a large grant from CIRM to study this approach, the intramuscular injection of vascular endothelial growth factor producing mesenchymal stem cells for the treatment of critical limb ischemia. So the goal will be to test this approach in a preclinical model, animal models, and then take it to the next step of a clinical trial in patients who are really at their wits' end and really down to their last option before needing an amputation for their severe peripheral artery disease. Vicki talked about some of the animal models for Huntington's disease. The animal model that we use to study peripheral artery disease is the hind limb ischemia model, where basically you take an animal, in this case a mouse, and you ligate the femoral artery, the main artery supplying blood flow to that leg. And then you can document the reduction in blood flow to that leg using a variety of techniques, including laser Doppler, as shown here, where we see now a reduction in flow to this right hind limb compared to the left hind limb. And once you've established this model of reduced blood flow, then you can test therapies that might subsequently increase or restore blood flow to that limb. If you inject these mice with stem cells, what you can see is that these stem cells go right away to where they're most needed, into that injured or ischemic limb. So this is the effect or the response that Jan has described so well as the paramedic response of the cells going immediately to where they're most needed. Now, if you then take those stem cells, engineer them to secrete a vascular growth factor, and then inject them into their veins, we can see a remarkable increase in blood flow within just a couple of weeks' time. As shown in this bar graph, the green bars just show that amount of increase in blood flow after injection of these engineered stem cells compared to placebo or just cells that do not secrete VEGF. And in this publication from Stem Cell in 2011, she showed proof of concept with this idea of the bioengineered mesenchymal stem cell, where after you ligate the femoral artery in the mouse and then inject these stem cells that engineer or secrete VEGF, we can see within 4 weeks a dramatic increase in perfusion in that right hind limb in that mouse, and shown graphically a remarkable increase in blood flow to that limb. And now in the more recent animal studies that Jan and her team have done, we see increase in blood flow to that limb extending out to 70 days after injection with the bioengineered mesenchymal stem cells. So an early and very prolonged increase in blood flow to the limb in these animals. So as Vicki talked about, the ultimate goal is to get these treatments into patients, into clinical trials in human beings with these very difficult problems. But you obviously have to go through the FDA first, and the FDA always has tremendous requirements in terms of the studies that need to be done to document the safety of the therapy, but also the efficacy. And these are just some of the trials that Jan and her team have been working so feverishly on over the past couple of years. And at the same time, we have been working very hard on the clinical trial, the protocol design, and what patients we're going to treat and how we're going to treat them. So the terminology is always interesting. Ultimately, you have to have a meeting with the FDA to talk about this investigational new drug or new treatment. But before you have the IND meeting, you have to have a pre-IND meeting. And before you have the pre-IND meeting, you have to have a pre-IND meeting, which in this case happened to be a conference call. So this is a photo from our pre-IND meeting with the FDA. And for that pre-IND meeting, we had to supply them with a 22-page document outlining our work and our plans for this experimental protocol. But the ultimate hope is that we will be entering clinical trials sometime next year, and we will be treating patients with so-called no-option critical limb ischemia, these patients that have such severe disease in their arteries in the legs and below the knee that angioplasty or surgery is really not a possibility. And without having something done, they're likely to go on and lose their limb, and the dreaded decrement in quality of life associated with the loss of that limb. So we're very hopeful that we will be able to make a significant change in the care of these patients to change their outlook, because we certainly know what will happen if we fail in our treatments for these folks. The dreaded below knee amputation on the right side, and the more dreaded above knee amputation on the left side, where this patient is probably never going to walk again because they're not going to be able to use a prosthesis. So thank you very much for your attention.
Thank you, that was great. And thank you all for staying, and I hope you've enjoyed seeing these three teams that I'm lucky enough to work with and be part of. And I think my job's the best one in the world because I get to go to all the team meetings and see what everybody's doing. And tonight you got to see what three of them are doing, and next year we'll have another three of our teams that are getting close to the clinic or maybe in the clinic. And just before we end, I'd like to give a special shout out to Gerald and Annette, who's going to be very embarrassed by this, but she is literally my right-hand woman, does everything in the program that I do, covers for me when I'm out or traveling, makes all these teams happen, does all the logistics. Thank you. Our unsung hero. And then also thanks to my amazing husband, Mark, who does everything at home so that I can work 18 to 20 hours a day on stem cells. And it's wonderful. Thank you, honey. I appreciate it. And we have a number of our students and members of the lab here. Everybody raise your hand who's in our lab. Yay, thank you guys. I appreciate it so much. And thank you to our amazing speakers. And it takes a village, and you're all a part of it. Thank you for coming and sharing and listening, and please come for a tour if you'd like to. Thank you.